Mariana Araújo. I'm a PhD student from ITQB, a research institute from Nova University of Lisbon, and my poster is focused on the adaptation of Staphylococcus aureus to atopic dermatitis skin lesion. Atopic dermatitis is a chronic inflammatory skin disorder, and Staphylococcus aureus have been associated with more severe AD forms, and the nose cavity being a potential source. So our objective was to confirm and to identify mechanisms associated with this adaptation. To accomplish this, we compare nose and skin lesion isolates collected from the same AD patients, and we perform several bioinformatic and phenotypic tests. So first, we sequence using both Illumina and technology our pairs, and we perform multi-locus sequence typing to understand the genetic background associated with AD. And we found that 60% of our pairs, so 32 pairs, belong to the exactly same sequence type. Among them, most belong to ST7, ST45, and ST15. After that, to identify phenotypic patterns associated with AD, we perform several phenotypic tests. So first, we compare the resistant profiles of the nose and the skin lesion isolates. And as we can see here, we have the number of pairs that lost resistance in the lesion identified with white and gain resistant in lesion identified with brown. And as we can see, most pairs lost resistant in the lesion, especially to antibiotics not used in the AD therapy, like for example, rifampicin and tetracycline. After that, in order to compare the virulence and the fitness of these isolates, we analyzed their ability to produce biofilm, growth rate, and urease production. And we found that the isolates from the skin have a higher ability to produce biofilms, a higher growth rate, and a higher urease production ability comparing with the nose isolates. After that, we tried to understand and to identify the genotypic factors associated with AD. So in here, I'm showing you the number of genes and plasmids absent in the lesion isolates identified with this brownish color and absent in the nose isolates identified with this pink color. We performed a genome alignment to identify deletions and insertions. And we found that the lesion isolates carried a low number of ribosomal RNA copies. After that, we performed a screening of resistant genes and virulence genes, and we found some differences, especially in antibiotics not used in atopic dermatitis. And we found that lesion isolates carried less adenosine genes. Finally, we compared the plasmids in these both in the nose and in the skin isolates, and we found also loss of plasmids in these pairs. Finally, we performed a SNP analysis to identify mutations. So in this graphic, I'm showing you the number of pairs and the number of mutations in each pair. We found between 1 to 33 mutations in each pair, with the exception of these two pairs that have a high number of mutation. Mostly are missense mutations. In this table, I'm showing you the genes affected by these missense mutations only and the functions associated, and mostly belong to genes associated with electron transport chain, virulence, especially adenosines, regulation, stress response, and cell wall membrane, lyase, replication and transcription, and biosynthesis of fatty acids, amino acids, and proteins. So to conclude, the adaptation to the AD skin appears to include loss of resistance to antibiotics, especially not associated with AD therapy, loss of ribosomal RNA genes, loss of plasmids, higher growth rates, alteration in virulence, in transcription, replication, and in the biosynthesis of proteins and amino acids, and finally increased biofilm of urease and production. Finally, I would like to acknowledge my supervisors and also FCT and NOVA for the funding and facility.